Alrighty, so I want to thank everybody for sticking around for the last talk today before we get into the closing keynote. And what I will be presenting on today is the importance of the source, composition, and bioavailability of dissolved organic matter in the York River Estuary. And it ties into my 6-word story, which is physics and biology control DOM dynamics. And that might not make sense now, but by the end of this presentation, I'm hoping that this makes more sense. So I want to begin with our York River watershed, which we've heard plenty about today. But I want to highlight that the York River watershed encompasses a variety of land types that influence the biogeochemistry of the York River. And what you're seeing here is a land map that shows the different land types that vary across the watershed. So even though most of the watershed is forested, as you see in the green, there are also some areas that are dominated by cropland and agriculture in the yellow, or developed land in the pink, as well as wetlands in the light blue. And as a result of this, these different land types all have some type of inputs into the aquatic environment, including that which is produced internally by the microorganisms within the system. So these inputs can come in the form of what we call dissolved organic matter, which is a term that describes the compounds that comprise organic carbon, nitrogen, and phosphorus that are all derived from these various sources. And it can kind of be grouped into organic matter derived from biological, geological, as well as anthropogenic sources. So why do we care about dissolved organic matter? Dissolved organic matter essentially forms the base of the aquatic food web because bacterial communities that do not photosynthesize harness their energy from using organic matter, particularly organic carbon. And you can see from this figure on the left that dissolved organic matter is incorporated into bacteria, where it can undergo several different fates, including direct respiration back into the atmosphere as carbon dioxide. It can be sequestered into bottom waters in the aquatic environment, and it can be converted back to their inorganic form of nutrients, as well as incorporated into higher trophic levels, as what we see here with the zooplankton. And therefore, investigating the cycling of dissolved organic matter in aquatic environments is important in order to assess the metabolic state of an estuary and how much carbon dioxide is being produced and released by this consumption. However, it's not really as simple as you might think. There are a suite of interacting factors that control the extent of how a DOM is cycled in the environment. And they can be kind of separated into biological, chemical, physical, and environmental factors. So we have biological factors such as the community composition of the microbes and bacteria that actually break down and consume the dissolved organic matter, and then associated rates of both production and respiration. We have chemical factors such as the source and the chemical composition of the dissolved organic matter. So certain compounds might be easier to break down and consume compared to others. And tying into that, the availability of nutrients also plays a role, going back to kind of a little bit about what Brianna talked about earlier today. And then we have physical factors such as water residence time, freshwater discharge, and tides, which essentially influence how long the organic matter stays within a system, especially in highly energetic places like estuaries. And then finally, we have other environmental variables such as salinity, dissolved oxygen, temperature, and light that you can kind of tie into all these other categories, but often these are a function of seasonality. So for the question I'm interested in looking at is, how does DOM consumption or bioavailability or degradation, I've been using those terms interchangeably, how does that change along the York River estuary under different environmental conditions? And I highlighted and bolded here the variables I will be considering with this talk. So respiration, the source and composition of the organic matter, as well as some physical aspects as well. So for this study, we considered several locations along the York River estuary and within its two main tributaries, the Pamunkey and Mattaponi rivers. So just to briefly go over these, we have the watershed fall lines at the Pamunkey and Mattaponi USGS gauging stations, as well as two tidal fresh sampling locations in either tributary, and then 3 sampling locations along the York River itself. And the station names along the York River were named just according to their relative location. So West Point is named just because there was a station closest to West Point, and so on and so forth. And I would like to note before moving on that stations in the green were only considered for DOM composition, so we could tease out the watershed contributions to the main stem of the York River, whereas the blue stations were considered for both composition and bioavailability. So going into a bit about our experimental design, first, I'll just mention that we did this study in October 2018, and then February, April, and July of 2019. And briefly, we collected surface water samples at each of those sampling sites where we filtered twice, once through a 0.7 micrometer filter, and then again through a 0.2 micrometer filter. The resulting filtrate from the 0.2 micrometer filter was then amended with the water that we filtered just through the 0.7 filter. And this allowed us to control for the volume of bacterial community that would be responsible for consuming the DOM. We then incubated these samples in the dark at ambient York River temperature, and then subsampled over the course of 2 to 4 weeks to measure changes in dissolved organic and inorganic forms of carbon and nitrogen, as well as chromophoric dissolved organic matter, which I will get into a bit, but that was used to give us an idea of the organic matter composition. So from these measurements over time, we calculated rates of organic matter decomposition. And I will note that we're using DOC as opposed to the entire organic matter pool because this is the largest fraction of the dissolved organic matter pool. And then we compared rates of those decompositions to the composition of the DOM itself, as well as those other physical, biological, and environmental factors. So before I move on, I want to briefly talk about how we use chromophoric dissolved organic matter for compositional analyses. And simply put, chromophoric dissolved organic matter is the fraction of organic matter that can absorb and fluoresce light. So within a given sample, that sample can absorb and fluoresce light at certain wavelengths along the visible spectrum of light, depending on its source and composition, producing what we call an excitation emission matrix, shown here. And you can see from the red color as opposed to the blue color that there is a peak intensity that occurs along certain wavelengths. And that peak intensity differs between samples according to its composition. So we have a 3-dimensional data set with all of these light measurements, but we can distill that down into different components based on how similar they are. So samples with similar peak intensities will be grouped together and assigned a component. And the number of components describes the composition of the organic matter pool in its entirety. So to kind of highlight how that relates to this study, for the York River estuary, I was able to identify 4 components that describe the composition of the organic matter pool during the time of the study. So identified a photorefractory terrestrial humic-like component, which means that this component is typically resistant to degradation. We have a photolabile component, which is an indication that degradation more easily occurs. We have a microbially derived humic-like, which indicates that degradation is occurring because the microbes are active. And then we have a soil fulvic-like component, which means there are contributions from soils. And when we look at the distribution of these components along the estuary, we found that the photorefractory pool was greater in abundance up estuary, especially during October and February. This was contrary to the photolabile pool, which was greater down estuary, and also in April and July. The microbial component was relatively constant across space and time, which makes sense if we assume that DOM is being consumed continuously. And then lastly, the soil fulvic-like component was consistently more abundant up estuary, which makes sense if we consider that this might have been from the watershed. And lastly, I will also note that there is a convergence of these components as you move down estuary, and that stations furthest down estuary were only comprised of these two components. And I'll get into that in a bit, and as far as diving into why we actually saw these differences in composition. So now what I want to show is how DOC reactivity or consumption changed across space and time. So this graph is showing you the percent reactivity or the percent of DOC that was lost over time. And the error bars here represent standard error of the mean of 3 replicates that were collected at each location in time. So positive values here are going to indicate that, for instance, 20% of DOC was lost, and then negative values would indicate that there could have been potentially a net accumulation of DOC. And then I will also note that the Pamunkey River location was only sampled in October and February, but we were still able to capture that up to down estuarine gradient moving near West Point down to the York River mouth. And overall from this graph, you can see that in October and February, DOC was most reactive up estuary compared to down estuary. And in fact, there was actually a net accumulation of DOC down estuary, which I'll get into in a bit of why I think that actually occurred. But the takeaway from at least October and February, about 18%, especially in the Pamunkey River, was lost over time. If we look at April and July, DOC reactivity actually shifted to being higher down estuary compared to up estuary. So we saw about a 20 to 30% loss of DOC near Claybank, as opposed to just about a 5% loss near West Point, and then 10% in April, 5% in July. So what could be explaining these patterns that we see? First, we can consider freshwater discharge. 2018 and part of 2019 were incredibly wet years. And this figure, Brianna showed the same exact figure before, which shows a plot of freshwater discharge into the York River. The red line indicates discharge during 2018 and 2019, while the gray bars represent the long-term average discharge between 1972 and 2017. So one possible explanation of our data is that in October and February, during these high discharge periods, the watershed could have been introducing more compositionally diverse DOC to the York River, which could have enhanced up estuary reactivity relative to downstream DOC reactivity. And again, in general, I'll mention that we did detect all 4 of those organic matter components up estuary, whereas we only detected 2 of those components down estuary. Secondly, we can look at the occurrence of a spring phytoplankton bloom. So in 2019, there was an intense bloom of the dinoflagellate heterocapsa that occurred mid to down estuary. And the approximate location of that bloom is indicated by the black star here. And this plot on your left is showing you evidence of this bloom using chlorophyll A concentrations that were measured along the estuary. And you can see that 2019 concentrations far exceeded the long-term average that's shown by the black line. So therefore, it's possible that in April, the phytoplankton bloom could have enhanced down estuary reactivity relative to up estuary reactivity due to the production of highly reactive or labile DOC. However, I do want to caution this notion because this is also the location of the secondary estuarine turbidity, which could also play a role in the high reactivity observed in either April or July. But that is something that was not considered for this presentation. So lastly, we can look at rates of respiration as a driving factor of DOC reactivity. So as a microbial community consumes DOC, it respires and also consumes oxygen as well in the process. So therefore, we could assume that high rates of oxygen consumption correlates to high DOC reactivity. So this plot is showing you exactly that. Here we have respiration in millimoles of oxygen per meter squared per day, moving down estuary between our different sampling dates. And negative values here are indicating a net consumption of oxygen. And what you can see is that in October and February, there's much less respiration occurring than what we saw in April and July, especially down estuary where we also saw those higher rates of respiration. So in conclusion, we can see that the reactivity of the DOC is dependent on these interacting biological, chemical, physical, and environmental factors. And I'm running out of time, so I just put up here the main conclusions to tie back to how physics and biology controls DOM dynamics in the system. But what I do want to end on is talking about what this means in the grand scheme of things. So the coastal landscape, including our Little York River, is susceptible to changes in climate and occurrence of disturbances such as storms, blooms, extreme discharge, warming temperatures, and the list goes on. And the point is that DOC reactivity could be susceptible to changes in environmental conditions, which could have implications for the metabolic state of estuaries. For instance, under conditions where DOC is rapidly consumed, estuaries could release more CO2 as heterotrophic processes could dominate. So this is also important for coastal carbon cycles and coastal carbon budgets in particular. So in the upper left-hand panel, this is a recent carbon budget for North American coastlines, showing you fluxes of carbon in teragrams of carbon per year, showing the exchange of carbon between different carbon pools. And if you focus in on the estuarine component, you see that estuaries outgas 10 plus or minus 10 teragrams of carbon per year, which is a 100% margin of error. So here I just want to highlight that if we don't even have a baseline understanding what's happening now, it will become more urgent if we want to figure out what might happen looking into the future under these different disturbance scenarios. So with that, I want to conclude by saying thank you to the funding agencies for this research, the National Science Foundation and the Virginia Water Resources Research Center, as well as several lab groups at VIMS. And thanks again for your attention, and I will be happy to answer questions through the chat.